What if you could feel this texture? This texture may give you pleasure or make you uneasy. However, by actively seeking the surface and exploring further through touch and other senses, you may be able to form a more coherent picture. Make a decision about how to interact with it. So we operate in the world through those active interactions with our environment, which are key for our daily lives and also our survival. What are the key mechanisms in our brain that enable this interaction between sensation and cognition during this active feedback loop? And most importantly, can a disruption between this active sensing and perception underlies cognitive impairment, like the one observed in neurodevelopmental disorder, such as the one associated with Syngap 1 haploinsufficiency? This is the burning question that major contributor for this project, JC, Sheldon, Gavin, and I, along with others, set out to address. So we were especially interested in this context of the role of Syngap 1, this postsynaptic protein that has been associated with a lot of major disorders, which include schizophrenia, epilepsy, and intellectual disability. But beyond its role at a synaptic level, we hypothesize that Syngap 1 impact circuit assembly and disrupt active sensory processes and perception, which are essential for normal cognition. So the main idea, if you like, is there's no proper cognition without proper perception and active sensing. And out of all the senses, we're really focused on touch, as it is one of really a fundamental process that occurs only on. And this was really striking in the case of Syngap 1, because thanks to a study published by Sheldon and the Syngap 1 patient registry, we had insight into the fact that touch-related sensory processing defects really were prevalent, as they constitute 35% of the sensory processing deficits that were described. So, in our case, we're especially interested in a mouse that can be used as a model for Syngap 1 haploinsufficiency. And past research from the lab have shown that behavioral deficits such as social interaction, learning, and memory are impaired, and there's a risk of anxiety and risk-taking behavior, which showed that this mouse model has good face validity. So our hypothesis was really that Syngap 1 mutation impacts circuit assembly, disrupting active sensing. But how do we go about studying this in the mouse system? So unlike us, mouse really probe their environment with their whiskers, and that's really what we use to as a surrogate for touch in the animal model. So I'm going to walk you through pieces of data that we gathered over the year and that really get to this idea by looking at first what happens when a stimulus is triggered and the response in a part of the brain, the somatosensory cortex, which is really fundamental to process this type of information. And now from there, we built a broader picture of the global circuits within the brain and identified key brain regions which we found are critical for the good active sensing that happens during more naturalistic tasks like pole presentation. Now this can impact the mouse behavior as a whole, exploring more complex environments. So initially what we found when we performed this type of piezo stimulation was that part of the brain where a reduced sensory-evoked brain activity. And that was quite interesting and very surprising because other parts of the brain in Syngap 1 and haploinsufficient mice have shown the opposite behavior, where in the mutant mice, what you would see is an hyper activity of the brain. So this suggested to us that these brains are way more complex and complexly organized. So we looked at the morphology and the structure at this one brain region especially, and throughout the development. And what we found was there is a decreased complexity in layer 2-3 and a strange dynamic in the layer 5, where the neuron seems to mature much faster in the mutant mice. So if you like, that's the type of trajectory throughout the development in terms of length and complexity of those neurons. And this really stemmed to us this idea that some early disruption may have like a critical impact on how those systems and circuits are built, not only at the place of the somatosensory cortex, but also more globally. So in order to address this, we focus not only on the structure, but also on the circuit as a whole. And in order to probe the circuit across the brain, what we did is we used a rabies virus strategy where you can inject a viral vector inside a specific brain area, and then it's going to spread across the brain and enable you to identify all the inputs which are received by the region of interest. And when we looked at the wild type and mutant mice, we didn't find any gross differences. And you can see an example of this type of quantification where most of the brain regions were found to be similar, except for one specific connectivity between the motor cortex and the somatosensory cortex. And this was confirmed through optogenetic experiment, where we could confirm this selective outside connectivity between those two brain areas. This now really led us to be like, okay, there's two key brain regions that seems to be disrupted within the circuit, especially S1, the somatosensory system, and M1, the motor system, which are essential for more naturalistic behavior, like active sensing when a mouse is presented with a pole and the way it explores it. And here you have example of the type of behavior that occurs in the mouse, where you can see that the wild-type mice are exploring the pole, touching, and the het mouse mutants do also to some extent. However, their exploration is much shorter. The interaction with the pole are much shorter. And you can see a quantification of this in the conventional knockout lines, where you can see this reduction. However, when we did the cortical knockout, you can see that this seems to be like cortical specific, and when we rescued Syngap in the cortex, this disappeared. So what happens now at the level of the circuit functionally? So now the idea was to be able to probe the brain and assess its function through different areas of electrodes across multiple brain areas. And while the mouse is performing the same task, now record the brain activity in the somatosensory cortex. And you can see that for each touch event, there is activity which are associated with S1. So now if we look at a quantification of that in the wild type, you can see the representation of different brain regions, like the thalamus for the upper panel, S1 for the middle panel, and motor for the bottom panel. And what you observe is consistent with what's been published with the literature. After the stimulus or like touch onset is presented as represented by the dashed line, you can see an increase in activity of S1 and an increase in activity of the thalamus as well, along with the motor cortex. Interestingly, however, the pattern is very different, with a striking reduction in the somatosensory cortex, and also a very different dynamic in terms of brain activity prior and after the touch onset. However, when we looked at the Whisking phenotype, although there is differences, they don't seem to be as drastic as with touch. So we really think this type of behavior, and we have additional data suggesting this, where the behavior during whisking is also normal, really relates to this disruption in the active sensing process itself, where you need to have sensory information and feedback occurring through this circuit that I was describing. So now we can hypothesize that this type of disruption will have a drastic impact on more naturalistic behavior, which are required for the mouse to perform, like textual discrimination. And what you can see is that there is indeed an impaired textual discrimination when two textures are presented. Then a wild-type animal is able to discriminate between the new object that is presented to it. However, a mutant cannot perform this type of behavior. So to us, taken all together, this piece of information really left us with this idea that Syngap mutation not only impact the circuit assembly, but also disrupt active sensing, which are required for cognitive processes. With that, I'd like to thank the National Institute of Health, my collaborator in the Miller and Rumble Lab, as well as our collaborator in Sweden, Dennis Medicine, Daniel Firth, and all the Syngap families for the insight they provide. Thank you very much for your attention. Enjoy the meeting.